Hi, my name is Margaret Gardel, and I run a biophysics lab at the University of Chicago. Today, I'm going to tell you about my favorite problem at the interface of biology and physics. When you first learn about cells, you think about the structure and function of individual organelles. What you learn much less about is the stuff that takes up the rest of the space within the cell, known as the cytoplasm. The cytoplasm is about 50% water. The rest of the volume is filled with densely packed globular proteins, small organelles, and dense filamentous networks known as the actin, microtubule, and intermediate filament cytoskeletons. So this complicated material can be thought of as a complex fluid or viscoelastic solid. The fact that you could call it either a liquid or a solid says that its mechanics depends on both the length and timescales you are considering. This field of soft condensed matter physics is well equipped to build physical models of such complicated materials. The fact that the cytoplasm is made out of macromolecules means that thermal effects are going to be important. If we now go down to the scale of individual proteins, in this animation, you can start to see the effects of thermal energy at this nanometer length scale. You see these randomized motions of individual macromolecules, known as Brownian motion, drives random agitation and collisions between macromolecules. You also get a sense of how crowded the cytoplasm is. All soft materials have this type of thermal effects at play. And so we built physical models of such soft materials in the physical world. What makes the cytoplasm unique is that it also contains an abundance of mechanoenzymes, that is, proteins that can convert chemical energy to do mechanical work. An excellent example of this is molecular motors. Here is an animation of a motor protein kinesin walking along a microtubule track. Right? The kinesin is taking in a chemical energy source and using that to do mechanical work, to drive its motility and generate local stresses. The fact that the cytoplasm contains all these mechanoenzymes means there's an external energy source in addition to just thermal effects. This drives the cytoplasm out of thermodynamic equilibrium and makes it a very unique material. So the cytoplasm is this soft material that is driven far from thermodynamic equilibrium. It has no analogs of similar types of materials in the physical world. This means that new physics is required to understand the physics of the cytoplasm. I'm going to talk briefly about two examples of where physics can play an important role in understanding aspects of cell physiology. The first is intracellular transport. For many different aspects, life processes in a cell, they have to move organelles and molecules from one side of the cell to the other. The types of cargo that need to be transported range from small molecules like water up to micron-sized organelles. Now, if we just think about thermal forces and diffusion, it's incapable of moving even the smallest cargo over very large distances in a reasonable time. Therefore, you need motor proteins to transport this cargo along the filamentous tracks. Now, if the cytoplasm were just simply water, the effective drag felt out by any of these cargo would be negligible, and motor proteins could do this quite easily. However, the cytoplasm is a very complex environment, and these cargo are going to feel very large resistances. How do motor proteins overcome this problem to really effectively move cargo? So if we just think about cargo moving in the cytoplasm, depending on your size, you're going to feel a different local environment. Small molecules are going to be able to diffuse rapidly and go in between the space of the globular proteins, whereas large cargo are going to be effectively trapped. This means that the type of material you're considering in the cytoplasm really depends on the length scale you're looking at. It'll be mostly liquid-like at small length scales, and it'll appear like a solid at larger length scales. This transition between a liquid and solid occurs at length scales relevant to organelles. Most proteins will experience a liquid-like environment, whereas chromosomes will experience a more solid-like environment. Now, how does a cell stay well mixed to be able to transport all of its objects and get them in the right place at the right time? Well, one recent idea is that the enzymatic activity within the cytoplasm actually serves to mix the cytoplasm up. This is akin to the advective mixing that you see if you put a pot of water on a stovetop, right? That advection really mixes the water in a way that's much better than diffusion. So this active stirring is thought to then liquidize the cytoplasm over all the relevant length scales so that chromosomes also feel a liquid-like environment. Now, the question is, does the enzyme activity really stir the cytoplasm, and is it possible that this enzymatic activity could be regulated to regulate the amount of stirring during different times of the cell metabolism? That is, could a cell effectively transition itself from being liquid-like to solid-like depending on its life stage? The second example I'd like to talk about is in cell morphogenesis. At the outer cell membrane, there's a dense network of filamentous proteins known as the actin cortex, and this actin cortex is responsible for cell shape changes that you see during cell division, cell migration, and tissue morphogenesis. So what is the mechanics of this outer polymer network that enables a cell to do these robust shape changes? So to get an idea of what this actin cortex looks like, you can see this fluorescence microscopy image of a reconstituted actin cortex where actin filaments are labeled in red. You can see that it is quite dense, over 100 micromolar, right? And it's a very thin, dense sheet of actin at the plasma membrane. And so what are the mechanics of this thin sheet? Well, actin networks are semi-flexible polymers. And what semi-flexible means is that their bending rigidity is comparable to thermal energy. And the ratio between these two components gives you a length scale that tells you the persistence length, the length scales of which the polymer maintains its rigidity. And since this length scale is 17 microns, that means an actin filament will sometimes behave flexible over larger length scales and behave more rigid over shorter length scales. Semi-flexible polymers are almost exclusively found in biological polymers and confer the networks that they form with very unique behaviors not seen in physical polymer networks. And this area of trying to understand how the semi-flexible affects force transmission is an active area of research. Secondly, we know that actin filaments are not static, but they're actively assembling and disassembling. And so how does this active assembly and disassembly of the polymers affect their mechanical response to the networks? We simply just don't have the models that integrate the polymerization dynamics with the mechanics. Some speculation is that polymerization dynamics are essential for fluidizing the cortex and enabling large shape changes while maintaining a uniform density during processes like cell division. So in addition to the actin filaments in the cortex, the main force generators in the cortex are myosin II motors. In this fluorescence image, myosin II is seen as an elongated rod. At the molecular scale, these elongated rods are actually bipolar filaments of myosin II motors that drive anti-parallel sliding of actin filaments. So these myosin II motors generate local stresses. In contrast to the highly organized sarcomeric structure of actomyosin that you see in striated myofibrils in muscle cells, in non-muscle cells, you can see actomyosin is very disorganized. This means that the myosin II is going to generate forces that vary in space and time. So we can start to explore how forces get transmitted in this very disorganized network by reconstituting again. These are actin filaments with myosin II motors on top of it, and we can start to see these random motions of myosin II. So because the actin cytoskeleton has so many, can be controlled spatially, we think that the architecture of the actin cytoskeleton is regulating the activity of the myosin II motors. But we simply lack the models to understand what is driving these types of responses. And likewise, you can see that the myosin II is forming these dense aggregates. This type of aggregation is not seen in wild-type cells. And so what in the cytoplasm is allowing myosin II contractile forces to not concentrate the cytoplasm in certain regions of the cell? So I've given you two examples of outstanding questions in cell mechanics. But I think there's even a greater challenge to be had at this interface. For instance, we currently now just build models that are specific to individual aspects of cell physiology. That is, we try to understand shape changes during cell division or cell morphogenesis or cell migration. But could we build a universal model of cell mechanics that guides a cell in all its different shape changes through its entire lifespan? Right? So can we really understand all the versatility in the cytoplasm that a cell experiences? Secondly, we have increasing knowledge about the molecular details of cellular processes. However, these molecular details do not confer the physical control parameters that are guiding the physics of the cytoplasm. So we don't have these control parameters, and currently different models confer different amounts of importance to different parameters. And so these need to be rectified, and we need to have predictive theories that then can be experimentally tested to see which one of these is correct. Hopefully, once we know the right control parameters, it will give us increased understanding of how the cytoplasm confers very robust responses in cells. That is, their ability to respond to a large range of environmental stimuli and undergo very dramatic shape changes. It may even tell us how these very complicated cytoplasmic structures evolved to give us such beautiful and beautiful mechanical responses. And so these and many other questions at the interface of cell biology and physics will give us lots of challenges for years to come.